Imagine imagine you're looking down at a massive, bustling metropolis.
Yeah.
Just millions of people, right? And almost every single one of them is texting.
Yeah, a city full of data.
Exactly. Now imagine you have this giant, incredibly powerful antenna, and you can intercept every single one of those text messages. You know exactly what is being said down to, like, the punctuation. But there's a major catch here. You have absolutely no idea where the senders or the receivers are actually located in the city.
Right. So you have the what, but you are, completely blind to the where. It's basically a logistical nightmare if you're trying to figure out how the city actually operates.
Yeah, and welcome to the deep dive, by the way. Because in the world of biology, that city texting nightmare was basically the state of affairs for years. We're talking about something called single-cell RNA sequencing.
Yes, the classic method.
Right, so scientists knew exactly what messages, like what genes our cells were using to communicate, but they just didn't have the GPS locations of those cells within the actual tissue.
You were just floating texts.
Exactly. Then this thing called spatial transcriptomics came along, which finally gave us a GPS map. But honestly, early on, it was kind of like a map that only showed large city blocks. It grouped cells into these clunky block-sized chunks.
Yeah, a neighborhood-level summary, basically.
Right. You still didn't know which specific house was texting which specific house. Which brings us to our mission today. We are doing a deep dive into this groundbreaking paper from Nature Genetics that fundamentally changes the resolution of that map.
It really does. It's a massive leap forward. Because a neighborhood-level summary just doesn't cut it when you're trying to understand the intricate networks that literally build a human brain or cause a complex disease. You need to see the individuals.
And that's why we're looking at this revolutionary computational tool today called CytoSignal.
CytoSignal, yes.
It is essentially the ultimate wiretap. CytoSignal tracks the exact cell-to-cell conversations, but at true single-cell resolution. It can distinguish between a cellular shout echoing across the street and, like, a secret handshake happening right next door.
It's moving us from a blurry, static photograph of cellular neighborhoods to a high-definition movie of individual cells talking to each other in real time.
And incredibly, by the time we get to the end of this deep dive, you'll see how it can even predict the future of those conversations. So, okay, let's unpack this.
Sounds good.
To really appreciate how this tool pulls off this magic trick, we first need to understand the biological hardware of a cell's communication, right? And the exact math that CytoSignal uses to score it. Because this isn't just taking pictures, it's crunching massive amounts of data.
Oh, absolutely. And it all boils down to essentially a lock and a key.
Okay, a lock and a key.
Right. So in cellular communication, the key, the message being sent, is a protein called a ligand.
Ligand, got it.
And the lock, or you could think of it as the inbox that sits on the receiving cell waiting for the message, that's a protein called a receptor.
So ligand is the key, receptor is the lock.
Exactly. When a ligand binds to the correct receptor, the signal is successfully transmitted, and the receiving cell changes its behavior. And CytoSignal's core mission is to calculate a mathematical score, which they call the LR score.
Ligand-receptor score.
Right, to quantify the exact strength of that specific interaction at every single spot in a tissue.
Now, I saw the formula for this LR score in the paper, and the underlying math seems to be simple multiplication, right? But the formula itself looked a bit more complicated with all these sub-numbers attached to it.
It looks intimidating, but at its core, yes, it is just multiplication. The interaction score, S, is the amount of ligand expressed multiplied by the amount of receptor expressed.
Okay, but why the sub-numbers then?
Because biology is rarely simple. Many of these signaling proteins aren't just single, solid pieces. There are complexes made up of multiple parts or subunits.
Oh, like a key that comes in 3 pieces you have to snap together?
Exactly.
Yeah.
So the exact algorithm calculates it like this. S equals the sum of ligand subunit 1 plus ligand subunit 2 multiplied by the sum of receptor subunit 1 plus receptor subunit 2.
So it adds up all the pieces of the key that the sending cell is producing, adds up all the pieces of the lock that the receiving cell is producing, and then multiplies those two totals together.
You got it. That gives you the total interaction strength.
Okay, that makes sense. I've actually been using a mailroom analogy to keep this straight in my head, paired with a mnemonic to remember the 2 totally different ways cells actually send these messages once they've printed them.
Oh, I like mnemonics. What is it?
Okay, so the first way is DED. Diffusible equals distance. These are ligands that are released into the wild, floating across the watery space of the tissue to reach their target.
Precisely. They physically drift. And to mathematically model these diffusible signals, CytoSignal uses a mechanism called the epsilon ball neighborhood.
The epsilon ball. Okay, what is that?
Imagine drawing a microscopic sphere, an epsilon ball, around a sending cell, extending out to a radius of about 200 micrometers.
Okay, so a 200 micrometer bubble around the cell.
Right. The algorithm assumes the cell can potentially receive diffusible ligands from any neighbor floating within that specific ball. It essentially sets a physical boundary on diffusion.
But wait, if a cell is shouting a diffusible ligand into the void, how does the algorithm prevent a distant cell on the very edge of that 200 micrometer ball from getting a false positive interaction score compared to a cell sitting right next door? I mean, in the real world, a shout gets quieter the further away you are.
That is a crucial point, and it's where the math gets really sophisticated. The algorithm doesn't just treat everything inside that 200 micrometer sphere equally.
Yeah.
It applies a distance weighting using something called a Gaussian kernel.
Hold on, Gaussian kernel. That sounds like a piece of hardware from a spacecraft manual. Are we basically just talking about a bell curve where the signal drops off a cliff the further away you get from the source?
That is exactly what it is. A Gaussian kernel in this context is just a mathematical curve that rapidly decays as the distance increases.
Oh,
So the physical distance dictates the weight of the signal. The sender cell's shout is mathematically muffled by the algorithm as it travels through the simulated tissue.
ensuring that distant cells aren't falsely scored as having a strong interaction.
Exactly. The math perfectly mimics the physical reality of a dissolving chemical trail.
That makes perfect sense. So that's DD, diffusible equals distance. The second type of communication is CC. Contact means close.
Yes, the direct touch.
Right, so these are protein keys and locks that are permanently stuck to the cell membrane. They cannot float away. The cells literally have to physically touch to communicate.
And to map these contact-dependent interactions, CytoSignal uses an entirely different geometric mechanism called Delaunay triangulation.
Okay, another big term. How does Delaunay triangulation work for physical contact?
It's a mathematical method used to draw a geometric web over a set of points. In this case, the points of the cells. The algorithm uses this triangulation to draw lines only between points that are immediately physically adjacent to one another.
I see.
It essentially maps out the shared borders, leaving out anyone who isn't a direct physical neighbor sharing a cell wall.
So it's like looking at a dense city from above and drawing a spider web that only connects the roofs of buildings that are physically leaning against each other. If there's even an alleyway between them, the web doesn't connect them, meaning they can't use contact signaling.
That's a perfect analogy. It cleanly separates the geometry of physical touch from the geometry of spatial diffusion.
Okay, let's do a quick mini recap of our first section here so we don't lose anyone in the weeds.
Good idea.
So bullet 1, cell communication is driven by these ligand-receptor pairs, basically biological locks and keys.
Correct.
Bullet 2, CytoSignal calculates an LR score by multiplying local ligand and receptor expression, adding up all their little subunits.
Yes, the basic math of the interaction.
Yeah. And bullet 3, it uses complex geometry, specifically Delaunay triangulation for physical contact, and epsilon balls with Gaussian kernels for diffusion to map the actual physics of the tissue.
Exactly.
So I throw a question to you, the listener. If you are a cell in an incredibly crowded, chaotic tissue, would you rely more on the secret handshake of physical contact to get your job done, or the broadcasting of diffusion?
It's a great thought experiment, really, because different cells absolutely choose different strategies based on their function.
Which leads us perfectly into section 2. How does this computational framework actually look when applied to a living, breathing map? Because they didn't just build this math in a vacuum, right?
No, not at all. They tested CytoSignal on embryonic mouse brains using complex spatial datasets known as slide-seq and slide tags.
Right. They wanted to see if CytoSignal could actually map the development of a brain while it's in the process of building itself. And they focused heavily on a specific interaction driven by a diffusible ligand called SMA3.
SMA3.
Which binds to a core receptor complex called plexin A4-NRP1.
This specific interaction is famous in neurobiology. It's absolutely essential for axon guidance.
Axon guidance.
Right. So when a brain is developing, nerve fibers, those are the axons they have to navigate through incredibly dense, complex tissue to find their correct targets.
Yeah.
That's how you eventually think and move.
Oh, they need a map.
Exactly. And SMA3 acts as a chemical breadcrumb trail guiding them. What CytoSignal revealed was a stunningly precise spatial gradient of this signal.
And what does that gradient actually look like on the map? I mean, visually.
Well, it doesn't just show that SMA3 was randomly scattered. It mapped exactly how the signal strength physically shifted across the layers of the brain tissue. It looks like a sweeping wave of color.
Wow.
Yeah, the signaling strength transiently increases, hits a massive concentrated peak, specifically in a dense layer of the brain called the subventricular zone, or SVZ, and then smoothly decreases as you move further away.
So you can literally see the communication hotspot, like the loud broadcasting tower, driving the brain's wiring in real time.
Exactly. And they contrasted that sweeping wash of color with other signaling behaviors. For example, they mapped 15A EFA7.
Okay, another alphabet soup one. What does that one do?
It's another diffusible signal crucial for patterning the embryonic cortex, which also showed a wide gradient. But then they mapped EFMB1 EFA4.
Let me guess, a contact-dependent one.
Bingo. It's a contact-dependent interaction known for its role in nerve regeneration. And on the map, the contact-dependent signals don't look like sweeping washes of color.
What do they look like?
They look incredibly sharp, pixelated, and hyper-localized to specific cellular borders. CytoSignal visualizes the stark difference between a neighborhood broadcasting a signal and cells whispering directly into their neighbor's ears.
That visual difference is amazing, but I have to play devil's advocate here for a second.
Go for it.
Knowing the message arrived at the lock is only half the battle, right? How do we know the cell actually read the text and changed its behavior? I mean, what if the key fit the lock, but the cell just ignored it?
That is the crucial next step. To prove the signal actually did something, you have to figure out which downstream genes inside the receiving cell were activated by that specific message.
Okay, so looking inside the cell.
Exactly. And to do this, the algorithm employs a statistical technique called sparse regression, specifically using what's called an elastic net model.
Okay, elastic net model. Are we trying to catch statistical fish here? Help me translate this.
In a way, yes. A cell has thousands of genes turning on and off constantly. It's incredibly noisy.
Right.
The elastic net model takes the interaction score we just calculated as the starting point, and it searches through the thousands of genes in the receiving cell to find the specific ones whose expression is directly mathematically triggered by that exact incoming signal.
Wow.
And crucially, it controls for the cell type as a confounder. Because some cell types are naturally louder or express certain genes by default, regardless of the incoming signal, the math filters out that background noise.
Okay, so the sparse regression is kind of like looking at a massive crowd doing a stadium wave. It's chaotic, everyone is moving, but this algorithm mathematically filters out everyone except the specific people who were triggered to stand up, only because the person immediately to their left handed them a note telling them to.
That is a fantastic way to put it, yes.
You're separating the actual causal chain reaction from just random people stretching their legs.
Exactly. And by separating the causal signals from the noise, they found that certain cell types act as distinct communication hubs.
Like who?
Fibroblasts, for instance, showed the highest levels of diffusion-dependent interactions. They are the broadcasters, scattering signals far and wide to organize tissue.
Okay.
Meanwhile, radial glia, which serve as physical scaffolding in the developing brain, showed massive amounts of contact-dependent signaling. They guide migrating cells through direct physical touch.
Okay, wow. Time for another mini recap.
Let's do it.
So, bullet one. CytoSignal maps physical gradients of signaling, like that massive peak of somathria in the brain's SVZ layer we just talked about.
Correct. Visualizing the broadcast.
Bullet two. Sparse regression identifies the specific downstream genes that are actually triggered by these signals, proving the message was read.
Filtering out the stadium noise.
Right. And bullet three. It separates the roles of different cell types, like the broadcasting fibroblasts and the whispering radial glia.
Spot on.
So to you listening, here's a question to chew on. How might understanding these exact physical gradients completely change the way we approach therapies for repairing nerve damage? I mean, could we synthetically mimic a 3D gradient to heal a spinal cord?
That is exactly the kind of therapeutic target this research opens up.
Yeah.
But we connect this back to the scientific process. Creating a beautiful computational map on a screen is impressive, sure.
Yeah.
But biology demands physical proof. A model is only as good as its physical validation.
Right, you can't just trust the math blindly.
Exactly. The researchers needed undeniable evidence that these microscopic, computationally predicted handshakes were actually happening in physical reality.
And this brings us to section 3, where the paper pulls out the heavy artillery. They needed a ground truth experiment, right? So they used something called the proximity ligation assay, or PLA.
The PLAs. Yeah.
And they performed this on Visium HD's spatial transcriptomic slides of a mouse embryo. But I have to admit, reading the methodology on PLA felt like reading a foreign language. How does this actually work in the lab to prove a computational score?
The mechanism of the proximity ligation assay is truly a marvel of bioengineering. Instead of looking at RNA, you're looking at the actual physical proteins in the tissue.
Okay, looking at the hardware itself.
Right. You introduce two primary antibodies into the tissue sample. One is engineered to bind exclusively to the specific ligand protein.
Pick a key.
And the other antibody binds exclusively to the receptor protein.
The lock. Okay, so you have these two flags attached to the lock and the key. How do we actually see the result?
This is the ingenious part. Attached to those primary antibodies are PLA probes, which are essentially tiny strands of DNA. You then add a special cocktail of enzymes. If and only if the ligand and the receptor are physically interacting, their attached DNA strands will be close enough to hybridize.
Hybridize meaning they connect.
Yes. They bind together, forming a continuous circle of DNA. An enzyme then amplifies that DNA circle, and you add fluorescent tags that bind to it.
So it literally lights up under a microscope.
Exactly. But here is the critical detail. Those DNA strands will only hybridize and create that fluorescent glow if the ligand and the receptor are within 40 nanometers of each other.
Wait, 40 nanometers?
Yes, 40.
Wow.
To put that in perspective, a single strand of human DNA is about 2.5 nanometers wide.
Okay.
A human hair is about 80,000 nanometers wide.
That is incredibly small.
This assay demands absolute microscopic physical intimacy between the proteins to produce a signal. It proves, physically and visually, that the key is currently inside the lock.
Okay, so the researchers took the interaction predictions made by the CytoSignal algorithm, and they benchmarked them against the actual glowing PLA dots under the microscope.
Yes, the ultimate test.
And they didn't just test their own tool, right? They tested older computational methods, too, like CellChat, spatial DM, and Slurn, and CytoSignal blew them out of the water. It achieved the highest area under the curve, or AUC, which is the statistical gold standard for predictive accuracy.
It really did. The accuracy was unprecedented.
But this is where I have to bring up a detail from the paper that genuinely made my jaw drop. The older cluster-based methods, they were predicting interactions between cells that were up to 8,000 micrometers apart.
Yes, 8,000.
Wait, 8,000 micrometers. The physical requirement for these proteins to interact is under 40 nanometers. That is a massive discrepancy. That is literally like saying two people are intimately holding hands when they're standing in completely different zip codes.
What's fascinating here is that this single experiment completely exposes the massive false positive rate of previous spatial methods.
It's terrifying, honestly.
Because the old tools grouped cells into those large block-sized clusters we talked about earlier, they just assumed that if a ligand was anywhere in cluster A and a receptor was anywhere in cluster B, they must be interacting.
Which PLA proved is totally wrong.
Completely physically wrong. It proved why CytoSignal's single-cell resolution isn't just a software upgrade. It's a massive paradigm shift in how we understand tissue biology.
Okay, time for recap number 3.
Let's hear it.
Bullet 1. PLA acts as the ultimate physical proof, fluorescing only when proteins are within that tiny 40 nanometer margin.
The undeniable truth.
Bullet 2. CytoSignal's predictions closely matched this physical reality, earning that high AUC score.
Yes.
And bullet 3. Older cluster-based methods produce massive false positive and false negative predictions, literally miles off biologically.
A huge reality check for the field.
Exactly. So listener, here's my question for you. Does knowing the high error rate of older computational methods make you look at past scientific studies with a more critical eye? I mean, how much of what we know is based on 8,000 micrometer handshakes?
It absolutely should make us read past literature with more critical eye. It's a sobering thought.
It really is.
But what's thrilling is that the researchers didn't stop at just proving the map was physically accurate for a single moment in time.
Right, because biology isn't a static photograph.
Exactly. Tissues are living, dynamic environments. The next frontier was figuring out how to predict where these signals are heading next. They wanted to turn the map into a movie.
Which brings us to section 4. Time travel, well, cellular time travel anyway. They developed a dynamic feature for this called VeloCytoSignal.
Yes, VeloCytoSignal. And it leverages a brilliant biological concept called RNA velocity.
RNA velocity, how does that work?
When a cell decides to manufacture a protein, it first creates a rough draft of the genetic instructions called unspliced RNA.
Unspliced, okay.
Then, molecular machinery edits those instructions down into a clean, final copy called mature spliced RNA.
So it's kind of like a factory floor moving from a rough prototype to a finished product on the assembly line.
Precisely. By comparing the ratio of unspliced, newly minted RNA to the mature spliced RNA inside a cell at any given moment, you can calculate the exact trajectory of a gene.
Oh, wow.
You can see if a cell is actively ramping up production of a signal or if it's in the process of shutting it off.
So if we apply that factory floor logic back to our ligands and receptors, VeloCytoSignal uses calculus to predict the future of a cellular conversation.
It does.
And the calculus formula they use is surprisingly logical, right? The instantaneous rate of change of the signaling, which they call DSDT, equals the change in ligand expression multiplied by the receptor plus the change in receptor expression multiplied by the ligand.
You nailed the math.
So we are literally drawing a weather map with wind arrows pointing toward increasing or decreasing signaling in a specific spatial spot?
Exactly. It draws a vector arrow showing the momentum of the conversation. And to prove the clinical value of this forecasting, they applied CytoSignal to a devastating disease model.
Right, the Parkinson's model.
Yes. Specifically, they looked at multiple samples of healthy young mice compared to old mice exhibiting Parkinson's-like pathology.
Now, the paper mentioned they used negative binomial regression to analyze this.
Yes, they did.
Whoa, another massive term. If we have young mice and old mice, why not just take a simple average of their signals and compare them? Why all the heavy statistics?
Because biological data, especially across multiple different animals, is incredibly chaotic and overdispersed.
Overdispersed.
Yeah, meaning the variance is huge. Some mice might naturally have more immune cells than others, completely independent of the disease.
Oh, I see.
Taking a simple average would bury the real disease signal in all that natural noise. Negative binomial regression is a specific statistical model designed to handle that chaotic, overdispersed count data.
So it filters the noise again?
It rigorously compares entire data sets like healthy brains versus diseased brains to isolate the exact communication lines that are fundamentally disrupted by the pathology, ignoring the natural biological variations between individual mice.
And that mathematical heavy lifting pays off because the findings they pulled out of that Parkinson's data were staggering. They found 42 diffusion-dependent and 3 contact-dependent interactions that completely shifted with age and disease.
A huge shift.
And specifically, immune-related signaling absolutely spiked in the older diseased mice. They mapped interactions with names like SPP1, CD44, and the SEMA60 plexin complex, and they were firing off constantly.
And that computational finding perfectly, elegantly matched the grim physical reality of the tissue.
What was happening in the tissue?
In those aging Parkinson's-like brains, there's a massive abnormal proliferation of microglial cells.
And those are
the primary immune cells of the central nervous system. CytoSignal tracked the exact dynamic communication networks those rogue immune cells were using to drive the brain inflammation.
So what does this all mean for us? Let's do our final recap of Section 4.
Perfect.
Bullet 1. VeloCytoSignal uses RNA velocity to calculate the future trajectory of cell signaling.
Yes, predicting the momentum.
Bullet 2. Negative binomial regression allows us to rigorously compare multiple samples, like healthy young versus diseased old, without getting lost in the noise.
Essential for real-world biology.
And bullet 3. In the Parkinson's model, CytoSignal successfully tracked the surge in immune signaling driven by those microglial cells.
Beautifully mapped.
So listener, if we can calculate the velocity of a disease's cellular communication, could we one day intercept the messages before the damage is permanent?
That is the ultimate profound promise of this technology.
It really is. So let's look at the arc of what we've covered today. We started with the raw math of ligand-receptor scores, right? Multiplying the subunits.
The foundation.
Moving on to map the sweeping 3D gradients of a developing brain, separating the broadcasters from the whisperers.
Yes, the visual maps.
Then we proved that math with the 40-nanometer physical truth of PLA fluorescence, completely exposing the massive flaws in older methods.
The ground truth.
And finally, we looked into the future of a Parkinson's disease state using VeloCytoSignal to watch the immune system actively shift its signaling in real time.
It's an incredible journey. From an abstract lock-and-key algorithm to a literal map of the future.
It really is. But before we wrap up, where do the creators of this text say they're heading next? Because I know this isn't the end of the road.
Oh, not at all. The authors of the paper are explicitly clear in their conclusion about their next target, and it is a vital one.
What is it?
The tumor microenvironment.
Oh, wow. Cancer.
Imagine applying VeloCytoSignal to cancer. We could watch in real time as malignant cells actively text surrounding healthy immune cells.
What would they be texting them?
Sending them diffusible signal to shut down, or tricking them through physical contact into actually protecting the tumor instead of fighting it.
That's terrifying, but also incredible to be able to see.
Right. And if we can map exactly where, how, and at what velocity those specific messages are being sent,
We can stop them.
we can use this tool to develop drugs that precisely cut the phone lines. We could blind the cancer and wake the immune system back up.
Cut the phone lines. I love that visual. Think about those trillions of text messages zipping around your own body right now as you listen to this. Your cells are shouting across neighborhoods, whispering to their neighbors, building scaffolding and defending against threats, all in a secret chemical language we are finally learning how to read. Keep exploring, keep questioning, and join us next time for another deep dive.